Okay, I think we can start now. It's time 11 a.m. Eastern Time. Welcome to the Vegetable Grafting webinar, Growing New Roots series. This is the monthly webinar we're gonna share. We are sharing the scientific information, the practical information around the technologies of vegetable grafting. I'm Cherry Kubota. I'm doing a host. Today's presentation is by Carrie Rivard, Kansas State University. Before introducing Carrie, as usual, I just wanted to mention about the International Symposium, the group of scientists organizing. Um, it's in Charlotte, North Carolina, from July 14th to 18th. There would be programs for industry people and then also extension people. So I would encourage to take a look at what we have in the program. I sent out the information the other day regarding program. If you would like more information, just let me know. It's gonna be interesting opportunity for you to learn and get connected with others. So with that, I'm going to introduce today's speaker. Dr. Carrie Rivard is from Kansas State University. He's currently associate professor at the Kansas State Research and Extension Center in Olathe. And he's also extension specialist and the director of that center. So he has many titles. Personally, I like his research program very much, very practical, focused, quite innovative. So I'm sure you enjoy his talk. And he's going to talk about grafting to increase production for small acreage and high tunnel tomato growers. So I'm going to stop sharing my slide, and then I will let, how do I do this? Sorry. Stop sharing. Here it is. I will let Carrie to take over. Sorry for my, all right.
No worries. Let's see if I can get this thing to work here.
Yeah.
Oh. Sorry, Jerry, I just lost you.
The green icon in the middle, bottom, in that.
You know, I use Zoom almost every day of my life. Somehow I lost my actual.
Yeah, so in the window of a whole bunch of videos, if you put the cursor down,
I found it. There we go. I got him.
Yeah.
Sorry about that all. I really do actually use Zoom quite a bit.
Mm-hmm.
I just got a new computer, so I'm trying to find all the buttons and everything to make it work. Okay, so can you see the screen okay?
Yes, beautiful.
Awesome. Well, thank you for that introduction, Sherry, and it's a pleasure to be here and to be invited to be a part of this program. I've been watching the webinars as well, and I've just been really excited about all the cool stuff that's been talked about through this webinar series, so I'm really excited to be a part of it. So I'm going to talk a little bit today about grafting for the use of high tunnel production, mainly for tomatoes. We do have a little experience grafting other vegetables. We just finished some work with peppers and have done a little bit of stuff with cucumbers, but not too much. Mainly we focus on tomatoes here at K-State University. And as Sherry mentioned, I am located at the Olathe Horticulture Center, which is basically a small research station just outside of Kansas City. And we're very fortunate to have a great group of folks that work there. We've got about 350 acres and a variety of graduate students and undergraduate students and community college students actually working here on all of our various projects. Now, one of the things that we have a real focus on here at the Olathe Horticulture is high tunnel production. High tunnels are really important for growers in Kansas. We have very difficult growing conditions. You know, we have a lot of wind, a lot of storms, not just the tornadoes we're known for, but on any given day, you can have a 20-mile-an-hour prevailing wind, especially down in central and south central Kansas. And so our growers are really rapidly adopting high tunnel technology, mainly as a way for environmental protection. And they do some season extension with it. What they're really trying to do is just protect their crops from the brutal state known as Kansas. So we've been building a lot of high tunnels out here at the Research Center. Some of these were here before I got here and were built by Dr. Ted Carey, who was my predecessor and did a lot of the pioneering work with high tunnels here in the Midwestern US. But we're fortunate in that we have quite a few different types of high tunnels, and it allows us to utilize these systems everywhere, anywhere from the most inexpensive homemade Caterpillar tunnel all the way up to the Cadillac model, solar-powered, 3-position movable tunnel that's on rails. So we've up to about an acre actually now of protected cultivation space. And although it makes for fun springs trying to get all these things covered, it does give us the ability to do lots of different types of research, which is one of the fun parts of my job. So if you're not familiar with high tunnel production, high tunnels are basically unheated passive thermal greenhouses. And if you're watching from outside the US, you may or may not know that the USDA and NRCS has been supporting the construction of high tunnels all over the country through an NRCS EQIP program. So our growers are actually able to get a cost share, oftentimes paying for more than 70% or even all of the high tunnel production construction costs, I should say, in order to help facilitate some local food production, especially around our cities, to help with local markets. Now, high tunnels are an extension of the plasticulture technology that started back in the 50s. The general idea is we want to grow under a covered structure. And one of the big differences between a greenhouse and a high tunnel is that in a high tunnel, we're typically growing in the soil that lays underneath of that structure, and rather than growing hydroponically. And this presents a lot of challenges in itself that I'll talk about here in a minute. Now, within Kansas, Missouri, Iowa, Nebraska, this whole Midwestern area, we have surveyed our growers, and it looks like about 82% of our growers are actually using high tunnels or want to, right? And actually, those numbers are a little bit old. My guess is it's probably higher than that. So we have, a lot of great farmers working in the area, and they don't always agree on everything. But one of the things they do tend to agree on is that the use of these covered structures really help their bottom line and help them be able to consistently bring goods to market throughout the course of the season. Now, tomatoes are the number one vegetable grown in high tunnels in the U.S. This has been reported in a couple of different studies. And for those of us that work in extension, we know as you travel around and visit these tunnels, it's very, very common to see tomatoes being grown in them. Obviously, high tunnels have a lot of benefits, right? And we've already talked about some of those in terms of extending the season. We also did a lot of work when I was a PhD student looking at reducing the prevalence of foliar diseases in organic high tunnels. And so just by the sheer fact of keeping the plants dry, you can really actually reduce a lot of those leaf spot and foliar issues that can be a problem from bacteria and fungi. But the main thing that our growers are seeing are just that increased yield and increased productivity. And Ted Carey did a lot of this work looking at open field versus high tunnel production back in the 2000s, and really showed that with lots of different vegetable and even some small fruit crops, increases in yield are pretty common to see when growing in the open field versus the tunnel. And again, especially in climates and environments like Kansas, where it's very difficult to grow in the open field. Now, all of that being said, there's a lot of challenges with growing in high tunnels as well, too. This is a picture taken from a farmer that I know, and you can see this is at the very end of the season, but this intensive production and oftentimes reduction in crop rotation intervals is leading to a lot of disease pressure for many of our growers within these high tunnels. What you're looking at now is a combination of Verticillium wilt, a little bit of botrytis, and some sclerotinia. And they're all kind of attacking at the end of the season and really reducing yields and causing issues. This grower, as it turns out, was actually growing tomatoes year after year after year without any crop rotation. And so as a plant pathologist, we know that's probably not the best thing to do. And in fact, what this grower has done since I took this picture, he's actually picked up his entire 2-acre high tunnel and moved it to another location on the farm because the prevalence of those soil-borne diseases was such a problem. So even though they're able to increase productivity, we do have a lot of challenges related to high tunnel production, especially with the lack of crop rotation and soil management in particular. excuse me. So as many of the people here probably already know, is that vegetable grafting is used worldwide, especially in high tunnel and protected cultivation systems. And it has been reported throughout the literature. We've seen that both in high-tech hydroponic greenhouses in Northern Europe, all the way to lower tech high tunnels and hoop houses that are around the Mediterranean region and Italy and Spain and the Morocco area, Northern Africa. Many of these folks are moving to using grafted plants. And so it seems likely that as growers in the US start to adopt high tunnels, as they start to get experienced in them and start to grow after a few years in that high tunnel, that they're probably going to start looking at the use of grafting as well. And as it turns out, this is one of the areas, again, where we do a lot of research. So, but worldwide, grafting has been used since the 1900s, and for tomatoes since the 1950s. So what are the benefits of using those grafted plants? I think a lot of the folks on this call already know that, but there's been numerous reports throughout the literature from all over the world about how you can manage soil-borne diseases. That rootstock is an entry point for a plant pathogen like a fungi, nematode, or bacteria, right? And so by utilizing a resistant rootstock, we can manage some of those soil-borne diseases that enter through the root system. And then there's a lot of issues related to abiotic stress, as well as other things like soil temperatures, drought stress, and other issues that have been reported on in the literature. So, we see that there's certainly potential for grafting to overcome some of those challenges that high tunnels present. But what are some of the challenges to actually using grafted plants, and especially here in the US where it's not quite as popular? One is certainly the cost. Within the last 5 years, there's been a number of grafted transplant nurseries that have popped up around the US and also throughout the country. I think there's a lot of smaller operations that are just producing them locally within a metro region for the Kansas City area or the Pennsylvania area as well too. So as we're starting to see those nurseries become more popular, I think the cost will come down. Excuse me, guys. But generally, there's a very low availability of plants in the US. And so, my hope is that as people realize the potential for this technology, I think we're going to start to see more and more nurseries pop up. And again, within the last 5 years, we've already seen some of that. Of course, one of the first questions from growers is, what is the actual economic feasibility of doing this? And we've reported some economic studies in the past. When I was working on my PhD, we found that it takes anywhere from 50 cents to $1 more to produce a grafted transplant, depending on how you do it, as compared to a non-grafted one. And so we can take that data and translate it to some of our yield data and look at the economic feasibility of doing this. And one of the things that we find is when we start looking across systems, so in this paper, and this was a paper led by Olya Reisen a few years ago, she basically showed that in a conventional open field system, you need an entire kilogram per plant of tomatoes in increase of yield in order to pay for the cost of the transplant. Whereas with some of our heirloom tomatoes and things grown in the high tunnel, those growers are selling at a much higher market price. And so they instead need 0.29 or 0.28 or 0.4 pounds of tomatoes in order to offset the cost of that added cost of using the grafted transplant. So this is where I think thinking about the system that you're growing in becomes really important. And it's not really that directly related to the high tunnel itself. It's more directly related to that most people that are growing in high tunnels are selling at a higher price than somebody in Florida who has a 30-acre field of high tunnel of tomatoes, excuse me. So if you're able to sell for $1 or more per pound, then suddenly it becomes really easy to afford a grafted transplant, whereas a very large-scale open field grower maybe is selling for 15 cents per pound, and it's really, really hard to make the margins that you need to afford that price for that additional transplant. So where are we at in the US? I'm seeing a lot of adoption of grafted plants within high tunnels. And again, this is one of the areas that we work quite a bit. But there's also been others that are really interested in using grafting, including home gardeners, some of our larger scale conventional farms, and some of our collaborators throughout the Southeast are really doing the research looking at that. And then, of course, heirloom growers, because heirlooms are susceptible to almost every known pathogen out there. And so if you can combine the great flavor of an heirloom tomato or some of these other specialty cultivars that are available with the disease resistance and vigor package of a rootstock, then suddenly you've got a plant that can really produce some money for you. So what about here in Kansas City? We've surveyed some of our growers, and we found that actually there's quite a bit of interest in using grafted plants. And we already have some growers that are using them, and actually these numbers are probably lower than they are currently. We're starting to see fairly rapid adoption of grafted plants. Now, of the people that are using grafted plants or interested in using grafted plants, there's still a very large number of our growers that want to graft their own transplants. And since we did this survey, I've learned a lot about the challenges of trying to get growers to produce their own grafted transplants. And for many of them, this just isn't simply possible. You do have to have a pretty nice greenhouse. You have to have the ability to manipulate that microclimate. Having cooling pads really makes the process much, much easier. And a lot of our growers just have very small, primitive greenhouses where they don't have the ability and the space to grow their own. And so, many of these prefer to purchase their own plants. So we see a growing market here in the Midwest for the use of grafted plants, again, that's following that market of the growth in high tunnel production. Now, I get a lot of questions from growers and other extension personnel about, can we actually use rootstocks as a replacement for rotation? And I showed you that picture earlier of that high tunnel that had a whole cadre of diseases associated with it because that grower had stopped using rotation, right? And usually when I get asked this question, as a plant pathologist, I would never recommend that somebody stops using crop rotation. In a soil-based system, rotation is so important for keeping those disease epidemics out of there and keeping, even if you have a little bit of disease, keeping it at bay so that it's manageable is really going to be reliant on utilizing that crop rotation. And so, we've actually done a lot of work in the last few years trying to develop successful crop rotations for our growers. Now, this is outside of the scope of this presentation, but I just wanted to point your attention towards this, that based on our data, it looks like there's a lot of what I would consider sort of medium value crops that can easily be grown in a high tunnel. And if we start thinking about the economics of it and looking at the economics of it, the grafting still plays a role because you can see basically that we have some sort of high dollar crops here, tomatoes at $3.66 and sweet potato slips at $4.30. And our strawberries are raising about $2 per square foot. And so those would be the ones that really push up that average annual gross income for the high tunnel grower. Even if you're not growing it every year, you can grow tomatoes 1 out of 6 years. And by adding that value and increasing the income during that particular year, it makes it easier to grow some of those medium value crops like brassicas and peppers and melons in the alternate years in order to establish that crop rotation. So it's okay to understand that the tomatoes are worth more, but thinking about revenue in the long term, they help to make it possible to afford to use a crop rotation. And by using grafted plants, we're increasing the diversity with that rootstock itself. But more importantly, if we can increase the productivity and the income associated with the tomatoes even more, then that makes it even easier to be able to bring in a little bit lower value in year 2 or year 4 in order to think about that revenue across the long term. Okay, so let's start talking specifically about the research that we're doing here related to tomato grafting. And I like to think of our grafting program as being separated into 4 key areas. We have done a lot of work in the past looking at different ways to grow transplants. As I mentioned, based on our survey data, our growers are very interested in growing their own grafted plants. And so we're interested in trying to find simple, effective methods that can be used in the greenhouse, in maybe a more primitive greenhouse than many of our commercial nurseries have, so they can grow their own plants. The other thing that we're doing all the time is we're always looking at new rootstocks, and we work closely with the seed companies in order to find out what's coming down the pike from our commercial seed companies from Europe and Asia, and what's going to be available in the U.S. and try and get a head start on knowing if that rootstock is going to be useful here in the Midwest. And I'll talk about that as we go too. And there's also a lot of projects that we've done that are related to high tunnel production and involving nutritional quality and rootstock and sign compatibility that I'll talk about. And then I'm hoping to finish up with just a little bit of information about some microbial ecology work that we did a couple of years ago that I find just extraordinarily cool. And it was really led by one of our collaborators in plant pathology. Okay, so let's talk about transplant propagation first. For tomatoes, we typically use what's known as the tube or splice grafting method, and there's been a lot of folks that have reported, provided publications and instructions how to do this. And this is obviously a very simple method where we graft below the cotyledon leaf of the rootstock, use a small silicone clip that slides onto that stem, insert the scion, and then we want that scion and rootstock to match up nice and tight so that we can have, as quick and as painless of a healing process as possible for that tomato. Once it's grafted, it goes into a healing chamber, and we've done a lot of some anecdotal and some replicated work just looking at different types of healing chambers that potentially a small acreage grower could use. And up in the top left, you can see these very simple what they call biodomes or humidity domes that you can buy from seed companies, and they actually work very well. If you're only trying to grow a couple 300 grafted plants, you don't necessarily have to put together this big fancy healing chamber with humidifiers and all that stuff. Now, the most important part of that healing chamber in that picture is actually not the dome, it's the styrofoam above it that's keeping the direct sun off of that little dome. It is really important anytime you're managing a healing chamber to prevent that double greenhouse effect, and it doesn't matter what scale you're working at. So I just wanted to point that out.
Carrie?
Yes, ma'am.
You can use the pointer if you
Oh,
want.
okay.
Yeah.
Thank you.
Your
That's
mouse.
great.
Yeah.
But inevitably, we've worked with a lot of growers that have tried to graft their own plants, and these are the types of pictures that generally get texted to my phone. Is pictures of a whole bunch of dead transplants, right? And that's the last thing we want. And there's a lot of reasons why that can happen. But so we've tried to do some work looking at different ways to manipulate that propagation process so that we can be more successful. So I've had a couple of graduate students that have worked on this, both Sarah Masterson and Lainey Meyer, and they both have graduated and gone on to do big things since then. One of the things that Sarah did was she actually looked at healing chamber coverings. And so we used, I think, 5 or 6 different treatments, anywhere from just standard plastic to only using shade cloth as a covering to using both. And we tried a few novel things as well, and we're able to publish that microclimate data a couple of years ago in Hort Technology. Now, one of the things that was interesting is although we had different environments that occurred within these healing chambers, the grafting success rate really wasn't affected by the actual healing chamber design itself. And some of that, I think, is a little bit of cockpit error. If you have a lot of practice grafting, especially with tomatoes, which isn't the hardest plant in the world to graft, then you just get high success rates. And maybe some of that was just luck for us, I'm not sure. But one of the things that I thought was interesting that came out of this work was that we were able to use leaf removal and shoot removal methods in order to reduce the, sorry, let me start that again. So we were able to use grafting methods that were successful in healing chambers with humidity levels that were lower than what you expect. So in this case, we had humidity healing chambers with humidity levels down around 50 to 60%, where basically we just use a piece of shade cloth as the healing chamber. And you can imagine for a small acreage grower that doesn't have a cooling pad, doesn't have the ability to block all the sun away from that chamber, this can be a really nice, simple method for them to be successful at grafting. And so, in our case, we did actually find that even with some of these chambers that had low relative humidity, we're able to be pretty successful grafting tomatoes. In terms of some of the methods that we've tried, Sarah played around both with shoot removal and leaf removal. Shoot removal was where we removed the entire scion meristem and everything and just left the cotyledons behind. And then leaf removal, we actually did different variations on a theme, as you'll see. But in most cases, what we're doing is trying to remove the leaves, but leave that growing point, that meristem, for the plant in the scion. And Sarah was actually able to find that although the healing chamber didn't have an effect on the success rate, the grafting method did actually, and we found that the use of that leaf removal was able to increase our grafting success by about 5%, moving it from 79 and 78 up to 84. So we were excited to see those results. And again, thinking about reducing the surface area of the scion makes it easier for a small acreage grower to manage that grafted plant in the healing chamber after they've gone through the grafting procedure. Now, one of the things that Sarah found with her work is that when we use shoot removal, and we didn't get a chance to test this with leaf removal, but when we use shoot removal, we did find that the yield was actually impacted negatively. So the time that it took for that transplant to regrow new shoots, new suckers, essentially, and then for those to become reproductive and do all their physiological things they need to do, set those plants back. And obviously, if you're a grower and you want to use that method, there's ways to offset that problem, earlier planting and those kinds of things. But if we just look at it as compared to a standard grafted plant, and this was with 2 rootstocks, in this case, Maxifort and Trooper Light, we found that it did actually delay some of those first harvests. So we're not really recommending people to use the shoot removal method, mainly because of that purpose. Now, I could definitely see for hydroponic growers, there is some functional use to using that shoot removal method. You're going to end up with 2 leaders that can be useful down the road. And I know a lot of folks that are propagating plants for greenhouse growers are pinching them anyways. And so you could actually get ahead of schedule in that case if you do it when you graft. But incidentally, for our high tunnel growers, it didn't seem to provide any benefit and actually was probably had a negative effect on the productivity. One of the other things that we've run into quite a bit, in addition to water stress in the healing chamber, is people that are new to grafting often actually over-humidify the healing chamber. And oftentimes they do this as a result of seeing wilting or other things. The greenhouse is really warm and they can't control the temperatures. But as a result, we oftentimes will see adventitious roots post-grafting. So these plants have already been healed, but they're in the final steps of that healing process. And suddenly, if the humidity levels are too high, they'll start shooting adventitious roots out from the scion. And that can be a real problem for many different reasons. We've seen adventitious roots get to the level where it will actually push the scion out of the clip. And so the graft union won't be successful anymore. Or in less severe issues, the graft will heal, but you now have an adventitious root from a susceptible scion going into the soil. And for high tunnel growers, this is a big problem because it creates a bridge for pathogens to enter that plant and potentially cause soil-borne diseases, right? So if you're grafting for the benefit of soil-borne disease management, you've lost all that benefit right there. And incidentally, we did this work a couple of years ago, but I was actually in Europe this past summer and took this picture this was at a very large-scale commercial production nursery. They had their healing chambers set up indoors, and they were using humidifiers and LED lights during the healing process. And as you can see, they had quite an issue related to adventitious roots. And so we talked a little bit about our research, and actually the guy who was operating the greenhouse had read our paper, so I was excited to hear that. But in their case, they were actually trimming these roots before they sent out. And this is a very large-scale nursery. They sell about 15 million grafted plants a year, and about half of those are tomatoes. So you can imagine, that can be a big economic problem as well. So Lainey Meyer was a graduate student that did quite a bit of work looking at adventitious rooting within the healing chamber. And one of the first things that we wondered about is, based on the literature, we knew that auxin production is not only related to the development of adventitious roots, but also originates from the leaves. And we had seen anecdotally that there did seem to be an effect on adventitious root formation as a result of leaf removal. And so she wanted to extend some of the work that Sarah had done to look at how leaf removal actually affects not only adventitious roots, but also the transplant quality and the growth and development of those transplants as they go forward. So you can see here on the bottom right, this was our standard grafted plants, and you can see lots of foliage there. Laney also had a 50% leaf removal, and you can see quite a few of those leaves have been removed, and then a 90% leaf removal. And you can see basically all that's left behind are the cotyledon leaves and just the stubs where the meristems were. Laney did a really nice job in her thesis and eventually publishing that work, and this is available through Scientia Horticulturae. And she did develop a scale where you can actually rate that adventitious root rating. And you can see from these pictures that we had adventitious roots that just got completely out of control if we let them. And so, again, you can see in some of these situations where you have like 8, 9, and 10, there's going to be a bridge here that goes down into potentially infested soils that can cause diseases, best case scenario. Worst case scenario, that plant can actually pop right off of the rootstock and become dependent on its own root system rather than the rootstock for nutrients, which obviously negates all the benefit of grafting altogether. So she did this experiment in several different healing chambers. This was one of them. This was a healing chamber that had high humidity. And basically what we found was that if we removed 90% of the leaves, we almost eliminated adventitious rooting altogether. And so you can see the categorized rankings and the proportion of the plants. And basically what this shows is that at 90% leaf removal, somewhere around 95% of our plants were what we considered acceptable according to this scale here, right? Whereas with 0% leaf removal, we only had about 45% that were considered acceptable. So that leaf removal makes a huge impact on the development of those adventitious roots. Laney also followed through this study with a greenhouse experiment, a pot experiment, looking at the plant growth. Now, we didn't take these all the way through to fruit production, but we did keep them in the greenhouse for long enough until they started flowering and setting fruit, because that was what we wanted to find out is really would it inhibit the earliness of these plants, right, in the time that they have to regrow. And what we found is that it did not. And in fact, what was kind of interesting is we actually saw slightly more pollinated flowers with our 50% leaf removal than we did with the non-grafted plants. Okay, so let's talk a little bit about our rootstock evaluation program. In years past, we've actually been a little overwhelmed with how much of this we were doing. There was a time when we would have upwards of 8 to 10 trials out in any given season, replicated trials looking at different rootstocks. Since in the last few years, we've actually reduced the amount of this that we're doing because so many of our growers are just interested in using the plants rather than experimenting with them now. And so we're trying to do our best to help our growers get their hands on the plants, but we are still doing rootstock trials as well. Here you can see a picture from a typical on-farm study. This is a Geringer's orchard, which is right around the corner from us at the research station. And this is one of the great things about my job is it gives me the opportunity to interact with growers and to do research on the farm. And here you can see 2 of their high tunnels. They have 7 of them, all of them over 100 feet long, and all of them full of grafted plants at this point. This is the first year we're not putting a study out at Geringer's because he told me, he's like, I just not really interested in having any non-grafted plants in my high tunnel anymore. So over the years, we've been able to collect a pretty nice list of plants that we consider able to increase productivity where there's little to no disease pressure evident within the high tunnel. And you can see the list on the left that's highlighted in yellow. These are some rootstocks that have pretty consistently increased in yield for us across many different trials in organic and conventional high tunnels. Now, we're generally using BHN 589 as a scion. We have done some work with heirlooms, but most of our growers are really interested in those red slicers that are growing in their high tunnels in this area. So most of it has been with the use of 589. I didn't want to put too much of this. We have piles and piles of data like this, but I just wanted to show you more of our recent data and some of the more up-to-date rootstocks that we've been working with. So Maxifort is a good old standard. Maxifort is probably the most commonly grown rootstock for high tunnel production in the US and in greenhouses as well. Multifort is sort of the sister of Maxifort, and my experience is that they behave very similarly. And then we have some other ones as well. Cardia, Fortamino is a pretty new one from Inzazaden, and we've seen pretty consistent results out of it for the last 3 years now. It also has pretty good nematode resistance, which is something we don't always get from the rootstocks, even when they say they do. And then Supernatural, of course, this is a new one that was just on the market last year. I'm probably wrong on this, but I don't think they're actually selling it this year. But it was touted as being one of the new up-and-coming rootstocks, and so we're really interested to see how it did. Now, the good news is there's still a lot of merit to grafting here, but the bad news is we still haven't found a rootstock that actually outproduces Maxifort. Typically, Maxifort is not only our good old standard, but it's also one of the highest yielding rootstocks that we utilize in any of our trials. Now, that being said, Colossus has done really well for us in the past, although it's off the market now. And I'm excited about Fortamino. I think it could potentially be really useful, especially for organic growers. This is some of the data. So that was some data from an on-farm trial at Geringer's. This is data from our research station. You can see a pretty similar trend, with the exception of the fact that Supernatural did a lot better at the Olathe Horticulture Center than it did at the on-farm trial. And we're not real sure why that is, but it's kind of interesting to see that there seems to be some kind of location interaction going on with that rootstock in particular. In this case at OREC, we also saw really good productivity out of Fortamino again, consistently, and this DRO 131, and that's actually that's a breeder's code. That's a rootstock available from DeRuyter. So as I mentioned before, we have piles and piles and piles of these rootstock trials that we've done since 2011 and even before that, for that matter. And so one of the things that we've been trying to do more recently is put that data together and think about it in a global way, so that we can try and look for trends across trials and across growing systems. And we're at a unique opportunity because we have all this data to really learn some powerful information, I think. And so I had a student working on this a couple of years ago, and this is David Lowen. He just finished his master's thesis and graduated this past winter. One of the things that David did was he actually did what many statisticians would call a meta-analysis, where you basically take all of our trial data from, I think in this case it was around 20 different replicated trials, and then try to find out trends that were happening across those trials. And so what these graphs represent, they are a little bit hard to quickly absorb, but what this basically means is the relative productivity on the left-hand side. So what that means is for example, if a rootstock had 25% higher yield than the non-grafted plant at any given trial, or any given replication actually in this case, then that means it has the relative productivity of 1.25, right? And then we did that as the y-axis, and then the production potential is the highest yielding treatment in the whole trial. So what that means is it's basically saying, okay, out of this trial, the best yield that we got per plant yield was maybe 30 pounds or 40 pounds per plant, right? And so what we kind of do is we can compare the general growing conditions with the relative productivity of the rootstock. And when we started doing this, we actually found some really interesting trends occurring. So Maxifort and Arnold, which are both considered vigorous rootstocks in my book, those are ones that consistently show yield benefits when there's little disease pressure, showed a positive correlation between production potential and relative productivity. So what that means is when you put those rootstocks in an environment where they're able to utilize their full genetic potential, they're able to do that. And you see that yield increase as the total yield of the productivity of the environment or the growing system increases. Whereas these two rootstocks on the bottom, RST04106 and RT1028, these are rootstocks that actually show the opposite trend. As the general growing conditions got better, and as we saw higher yields in that trial or in that environment, the relative benefit of grafting went down. And that's probably a result of the non-grafted plants doing better rather than the grafted plants doing worse. But either way, what it shows is that we don't have that trend. So in this case, we're not able to increase the genetic yield potential of the plant, right? And so what we're trying to do now is take some of this data, and I'm really interested in continuing and doing more of this work and thinking about how we define a vigorous rootstock. Is it one that increases its yield as that environment gets better, or is it one that just adds 10 pounds onto every plant and stops there, right? So, there's a lot of fun stuff that we can do with this data, but we've just begun to really crack the surface of this. So to kind of wrap things up with our rootstocks, again, we're really looking at rootstocks that can increase yield and productivity in situations where there's little disease pressure. And this is kind of an old table here that we developed a few years ago at NC State. But what we're trying to do is add another column to it, right? And think about the vigor and really the vegetative tendency of those plants and their ability to increase yield when there's little disease pressure. And what we've been able to do is identify a couple of rootstocks like Maxifort, Multifort, and Colossus that really increase the yield even when there's little disease pressure available. So let's talk a little bit about some of our high tunnel, general high tunnel production experiments as well. And I think this project really ties nicely into that last slide, because at some point, one of the things that we learned or occurred to us was, we look at all these different rootstocks and see how they affect yield, but we've never looked at scion, right, and how that affects that per plant productivity. So this is another part of David Lowen's master's thesis, and what he did was he actually grafted 10 different scions onto Maxifort rootstock, and then we looked at the marketable and total yield. And now it's not as interesting to look at here, but if you normalize that data into percents, then you can see a striking difference between the scion compatibility of certain varieties compared to others. So for example, what we see here is that BHN 589, Red Deuce, Skyway, and Tasty Lee are all increasing their percent yield by anywhere from 40 to 60%, right? So that's a really nice increase in yield for a grower. And that's something that if you're paying an extra buck per plant, it's probably going to be worth it, right? Whereas Primo Red, look at this guy, the increase in yield was something like 9%, right? And so in that case, the increase in yield or the benefit of using this vigorous vegetative rootstock just simply wasn't there. And it wasn't because there was anything wrong with the rootstock. It was because the scion, for one, did really well as a non-grafted plant. And number two, it wasn't able to utilize that energy from that vegetative rootstock. Now, David looked at this in a few different ways because one of the things that we're trying to get at is, having the long-term vision of having lists of scion that are ranked into different orders of compatibility. Okay, so one of the things that he did was he first ranked them as a non-grafted plant, highest to lowest, right? And you can see this on the left-hand side. So Primo Red was one of our highest yielding non-grafted plants. And then once it became grafted with Maxifort, how did that list change? So if we think about the function of a rootstock, if this is an additive function, and if we think a rootstock is going to add 10 pounds per plant onto every scion that we would graft with, then that order, that ranking should be the exact same. But in fact, that's not the case at all. What happened was certain rootstocks it added a lot to, others not so much, and so we suddenly see the ranking and the performance of those scion varieties, or really those grafted plants, very different than the non-grafted ones. So what's nice about David's work is he was able to make a list of some of these really compatible varieties that tend to increase, that tend to utilize the benefit of those vegetative rootstocks more efficiently than maybe some of the other ones do. And you can see there's a lot of fuzzy area between moderately compatible and highly compatible. At some point, we just had to pick a number, and so 40% was it. But there's also, I think, what's even more important for growers is this list of compatible plants at the bottom, right? Scarlet Red, Primo Red, Red Morning. Those are plants, if you're interested in growing those varieties in your high tunnel, I probably wouldn't recommend using grafted plants because we just don't really see the benefit of using that expensive rootstock in order to increase the yield. And I think some of it is because they're pretty high yielding non-grafted plants, and some of it is probably because they're not able to balance that extra energy that the rootstock gives. So we're going to shift gears a little bit now and talk about fruit quality. And I don't have a lot here. I've done just a small amount of this work. I know some of my collaborators in Washington and in Florida have also done much of this work as well too. But we've been working closely with Dr. Eleni Pliaconi, who is a faculty member here at K-State, and she runs our post-harvest physiology lab at the K-State Olathe campus. And so we have a nice opportunity, or we have over the years with all these rootstock trials going on, collect some fruit from there and see how the yield, not only the yield is affected, but also the nutritional organoleptic quality. So here you can see some of that data, and this is 3 years of data combined with several different rootstocks, and these were plants that were grown at our research station. We did also sample fruit from some of the on-farm trials as well, and saw pretty similar data. But basically what we found out is there wasn't any effect on the Brix levels of the grafted versus the non-grafted plant, and it didn't really matter what rootstock they were grafted with. But we did find actually a little bit of significant difference within titratable acidity. So the plants that were grafted with Maxifor rootstock were actually slightly more acidic than the non-grafted plants and the ones grafted onto RT1028 and RSTO4106. Now keep in mind that those last two rootstocks are ones that we consider to generally not increase the yield of our plants. And in this case, I can tell you from the yield data that we didn't see any increases in yield with those either. But the high yielding variety Maxifor, we did see slightly more acidity. When you talk to post-harvest physiologists, and I do that a lot, about flavor, one of the things that's really important for tomatoes is the ratio of sugars to acids, right? And that's, as a human being, that's one of the ways we interpret flavor, is that ratio of sugars to acids, right? And so the higher acidity fruit will affect the flavor some, and it's something we need to keep an eye out for. Now, if we look at nutritional quality, and in this case, antioxidant capacity, we did see a little bit of difference, but it's probably not what you would expect. There's a general assumption out there in agriculture that as we increase yield with any given crop, we're going to lose nutritional quality. And I think there's a lot of people that are interested in heirloom varieties and some of those other things because of that general trend that I think in many cases is an assumption. But we wanted to see, if that was the case here. So what we found is that when we measure antioxidant capacities in ORAC, which is just one of the methods they use in the lab, we didn't see any difference. But when we tested it using FRAP, we actually did. Now in this case, the high yielding variety, which was Maxifor, had similar nutritional quality, similar antioxidant capacity to our non-grafted plants. What was interesting was that our low-yielding varieties, RT1028 and RSTO4106, actually had higher antioxidant capacity than the non-grafted plants or the ones grafted with Maxifor. So that's a really interesting phenomenon. We've actually been following up on it in the last 2 years, and I'm not going to get a chance to show that data, but we're working with a high lycopene variety called Tasty Lee, where we're hoping to actually see if there's a reason to use some of these low-yielding rootstocks. Okay, just to kind of wrap things up now, I just wanted to mention some of the work that we're doing, look at microbial ecology. When you think about rootstocks, many of the rootstocks that we're using are interspecific hybrids. So they're going to have a cross between a wild and a domestic species. So it makes sense. Logically, you could see that it would be likely that the microbial communities that are associated with those root systems could be different, right? And so we had a student who's just finishing his PhD now. He actually started here at K-State, and then Karen Garrett, his supervisor, moved to Florida, and so they've just finished it up now, and we're getting this. We just had the first paper published, and we're hoping to get the next one published fairly soon. And what Robin was able to do was look at both the endosphere, in other words, inside the root, as well as the rhizosphere, which is the surface of the root. And using genomics, he was able to identify some of the different bacteria and fungi that were present on those plants. And this is just the bacteria study here. He also has a whole nother list of fungi as well that are associated with these plant roots. But we did find that the abundance of some of these particular groups was affected by using rootstocks as well as self-grafting. And the other thing that was interesting was that the diversity of the microbial community was also affected by these vegetative rootstocks. So Maxifort actually had a higher diversity index within the rhizosphere as well as the endosphere than the non-grafted plants and those that have low yielding plants like RST04 106. So, you know, it kind of makes sense if you think about it. Many of these vegetative rootstocks, we know that they generally have a larger shoot biomass, the plant does, and so it's going to be able to send more photosynthate and more food down to the root system, which could support a different microbial ecosystem on the rhizosphere or within the plant roots themselves. And last but not least, Robin was also able to identify some of the specific players that were associated with Maxifort, rootstock, and some of these other non-grafted and self-grafted controls. So I'm not going to get into the details of that, but that paper was just published a few months ago, and if anybody would like to get a copy of it, feel free to reach out to me. Okay, so just to kind of wrap things up, you know, my observations is that it really looks like grafting is here to stay for high tunnel growers in the US. I think the verdict is still out a little bit for many of our large-scale open field growers as to whether or not the economics really are going to make sense for a 50-acre farmer in Florida to, you know, grow using grafted plants. But for those of us that are working in small spaces and trying to push the dollars per square foot and within a high tunnel, then it generally makes economic sense. And again, a lot of this has less to do with the grafting technology and just the sheer coincidence that people that are growing in high tunnels are generally getting a higher market price for those tomatoes and are generally cultivar focused. They want to grow specialty cultivars or heirloom cultivars that they know are going to do well in their high tunnel. We've also found that the productivity of grafted plants in high tunnels is generally higher than non-grafted plants. And that's with certain rootstocks like Maxifort, Arnold, Colossus, and now we're seeing Fortamino also has pretty good results. But what I think is more interesting is just this compatibility thing that we've been able to investigate looking at scion and rootstock compatibility, because it does seem like that's actually probably more important than was previously speculated by myself and other researchers around the country. As far as the quality issues, you know, again, it looks like we still need some more data. But it does seem like the organoleptic quality is minimally affected, and in particular the acidity with certain rootstocks. But you know, we haven't done any consumer testing yet here in Kansas. I know some has been done in Florida. So I think that's really the important part of that research, is to determine if that quality is noticeable by a human being, right? And then of course, it does seem like our microbial communities are affected. But more research is needed. The work that we did and Robin did, I should say, was really very much like survey work. Say, is this phenomenon even happening? And now the future is wide open for what we can do with that information. Can we start breeding rootstocks for high microbial diversity? Can we breed rootstocks in order to try and attract certain microbes that have beneficial aspects to them? You know, it's a black box in terms of what we can do with that information. So again, we're just excited to get it out there. But you know, overall, what I'm seeing is the practical application for our farmers is very real. And this is a picture. This is Dan Kuhn, who's a farmer here in Cortland, Kansas, and Dan has got a whole high tunnel full of grafted tomatoes behind him. And you know, and I've heard this from him as well as other growers, and they've maxed out their productivity within the high tunnel, and they say, why would I build another high tunnel when I can just use grafted plants? And I have no real answer for that other than making sure that I want to make sure they use crop rotation and use those other, you know, good IPM practices. You know, I think that logic makes a lot of sense for many of these growers. So we're starting to see really rapid adoption of this technology in Kansas and Missouri and throughout the Midwest. And I would expect that you're going to see that as well throughout the rest of the country. Okay, I think I've taken a little more time than I meant to, because I do want to have a minute for questions at least, but there's a lot of acknowledgments I'd like to put out, mainly to our graduate students and technicians that collect all this data. Any of this applied research, it takes a lot of physical work, right? And so there's a lot of people that have helped along the way, and I really want to acknowledge that, as well as our funders, the Specialty Crop Research Initiative within NIFA, as well as some of the other funders that have supported our work. And with that, I would be happy to take any questions, and I thank you for your attention.
Well, thank you so much, Carrie. It's quite interesting sharing different project results and outcomes. Okay, so this is the time for you to answer questions. We have 5 minutes or so, and you can do two ways. One is just turn on your microphone and speak up. I like that way because I don't have to read your comment in the chat area, but if you prefer to write in the chat area, you can do that too. So question? Anyone? So I have a question to start. So the quality change by grafting, it was quite interesting. You saw acidity or the titratable acidity reduced by some of the rootstocks. Have you seen any difference in terms of environmental disorders such as blossom end rot between grafted and non-grafted?
Yeah, that's a great question. And blossom end rot can be pretty problematic for our high tunnel growers in particular. Back in the day, and this has been several years ago when I was in North Carolina, we had a couple of years where we saw lower blossom end rot incidence with the use of Beaufort rootstock. Not so much Maxifort, but Beaufort. And Beaufort's been off the market now for a little while. But certainly you can see how its ability to bring in more water and more calcium could certainly affect the incidence. So
yeah.
we haven't been tracking that in recent years specifically, but I could certainly see as it being a useful thing to look at in the future.
Yeah. Another question while people are trying to form their questions. What do you think, in the survey you showed, majority of high tunnel growers or small growers want to graft if they can do. What do you think is the amount they can easily graft? Is that going to be 1,000 plants, or is that going to be 5,000 or 10,000? What do you think in your experience and working with growers?
Yeah, it all just depends on what kind of greenhouse they have. I mean, that's what it comes down to. And, to be honest with you, that survey data, there was a lot of people that answered they want to produce their own grafted transplants that have never tried to produce their own grafted transplants. In the last couple of years, we've started doing custom grafting for our farmers here in the area, and have gotten quite a response. There's actually a lot of people that want to buy grafted transplants, mainly because they just don't have the facilities to do it themselves.
Right.
But in terms of numbers, I think of somebody that would be a small batch grafter as somebody that wants to grow 1,000 plants or less. And then the next scale is sort of 5,000 plants, 5,000 to 10,000 plants you can do, in a medium-sized greenhouse with a small healing chamber facility.
Yeah. Okay, good. All right. So
yes, I have one question.
Go ahead.
Yes, related to the leaf removal technique, I'm wondering if what would be better if we remove the leaves, or another technique that is to decrease the humidity in the time that plants are in the healing chamber. I mean, this is because the labor cost of and it's the extra time that we need to remove the leaves.
Right. So you're talking about for adventitious rooting problems in particular?
Yes.
Yeah. No, I would agree. I think as you get better at growing grafted plants, you learn to walk that line of the exact right humidity level you need that's not too high and not too low. But many of our folks around here that are trying it for themselves, it's maybe their first time, or maybe they tried it last year and they were a miserable failure and they want to do it again this year. And so they tend to be folks that increase the humidity way too much in their healing chamber, right? Because they're just worried about the plants wilting, or we've one of the things we've noticed over the years is about the 5th day after grafting, the plants are going to wilt, right? As they're reconnecting that vascular tissue. And if you don't have that experience and you don't know that they're going to pop out of it the next day, then your tendency is to just crank up the humidifier and, you know, hope that the plants come back to life. And that's the situation where we see these mega adventitious roots pop out, is because they don't understand that, because they're new to grafting, they don't understand those plants are going to wilt a little bit anyway, and then they'll pop out of it the next day. So, you know, I think in terms of your question, if you're able to do it successfully at lower humidity, that certainly would be a better way to go. But for many of our new grafters, they're just not able to walk that line as efficiently.
Okay, thank you.
Anyone else?
Looked like there was a question on the chat box. I think from somebody I know. So there's a question about rootstock germination and growing, damping off longer stems. Yeah, there's a whole bunch of questions there. So I don't have any specific data available on rootstock germination. One of the things that we find is sometimes we'll get rootstock in and it germinates at 70%, and sometimes we get rootstock in and it germinates at 98%. And it can be the exact same variety from the exact same seed company. Now, I know a lot of the seed companies are using priming as a way to increase the germination rates within rootstock specifically, but it seems like maybe some of them do it better than others, or maybe different batches have a you know, different efficacy. So even if you had data on rootstock germination, you're going to find that it's quite variable on both sides of the spectrum. In terms of damping off, I've never had an issue with any of our rootstocks damping off, but I go to great lengths to keep damping off out of my greenhouses in general. So I haven't really tested them by inoculating them or anything like that. And in terms of longer stems, we don't do any light manipulation to lengthen the stems, although my understanding is that some nurseries will do this. One thing that we try and be real careful about is when we transplant the seedlings, we don't want to plant them too deep, because we want to make sure those cotyledon leaves are as high off the soil line as possible. So that's one of the things that we do, but we haven't really played around with lighting or some of these other methods, and Chiri would probably know more about that than myself.
Any other questions? Anyone else?
Yes, I have one. For the high tunnels, you're putting the plants out earlier, which means you got to get your plants started earlier. And my question is, how much does the length of day impact that, as opposed to just sunlight? Even if you have a greenhouse facility, you just don't have a length of day in order to get the plants started early enough for early greenhouse.
Yeah, so once we got into the custom grafting world, we figured out that many of our farmers want transplants earlier for their high tunnels than we actually plant them in our high tunnels, yes. Which works out fine for us because we get it all done before we start grafting for our research trials. So I definitely recognize the importance of that. What we tend to estimate that it takes us 8 weeks to grow a grafted plant from seed to hardening during January, February, and March. And once you get up to March, April, and May, it's more like 6 to 7 weeks. Kind of depends, again, on the weather. But that phenomenon of shorter day length definitely makes an effect on how long it takes to grow that plant, for sure.
Okay. So I guess it's already a little bit over the original time. So I would like to conclude the May webinar on vegetable grafting. And then thank you, Carrie, again, so much for having your time and then sharing your information with us. Thank you, everyone else, being here, participating in this webinar series. So more information coming up in the future, and then look forward to that. Thank you so much. Have a good day or morning, whatever you have. Thank you.
Thanks, everybody.